Natural excitation of the heart. The nervous system controls various aspects of cardiac behavior, such as the heart rate and the strength of each contraction. However, cardiac function does not require an intact innervation. Indeed, the ability of heart function and adapt to changing conditions lies in certain intrinsic properties of cardiac tissue, especially its automaticity. The properties of automaticity, that is the ability to initiate its own beat, and rhythmicity, that is the regularity of such pacemaking activity, allows the heart to beat even when it is completely removed from the body. If the coronary vasculature of an heart is artificially perfused with blood or an oxygenated electrolyte solution, rhythmic cardiac contraction persists for many hours. At least some cell in the atria and ventricles can initiate beats. Such cells mainly reside in the nodal tissues or a specialized conducting fiber of the heart. The region of the mammalian heart that ordinarily generates impulses at the greatest frequency is the sinoatrial node, SA node. It is the main pacemaker of the heart. Detailed mapping of the electrical potential on the surface of the right atrium reveals that 2 or 3 sites of automaticity located 1 or 2 cm from the SA node itself serve along with the SA node as an atrial pacemaker complex. Region of the heart other than the SA node may initiate beats under special circumstances. Such sites are called ectopic foci or ectopic pacemakers. Ectopic foci may become pacemakers when: first, their own rhythmicity becomes enhanced. Second, the rhythmicity of the higher order pacemakers become depressed. Or third, all conduction pathways between the ectopic focus and those regions with greater rhythmicity become blocked. Ectopic pacemakers may act as a safety mechanism when normal pacemaking centers fail. However, if an ectopic center fires while the normal pacemaking center still functions, the ectopic activity may induce either sporadic rhythm disturbances such as premature depolarizations or continuous rhythmic disturbances such as paroxysmal tachycardias. When the SA node or other component of the atrial pacemaker complex are excised or destroyed, pacemaker cells in the AV junction usually take over the pacemaker function for the entire heart. Sinoatrial node. In human, the SA node is about 8 mm long and 2 mm thick and lies posteriorly in the groove at the junction between the superior vena cava and the right atrium. The SA node contains 2 principal types of cells. First, small round cells which have few organelles and myofibrils, and second, slender elongated cells which are intermediate in appearance between the round and the ordinary atrial myocardial cells. The round cells are probably the pacemaker cells, whereas the slender elongated cell probably conduct the impulses within the node and to the nodal margin. Atrial conduction. From the SA node, the cardiac impulse spread rapidly throughout the right atrium along ordinary atrial myocardial fibers at a conduction velocity of approximately 1 meter per second. There are 3 internodal pathways between SA node and AV node: anterior tract of Bachmann, middle tract of Wenckebach, and posterior tract of Thorel. A special pathway, the anterior interatrial myocardial band or Bachmann bundle, conduct the impulse from the SA node directly to the left atrium. The wave of excitation that proceeds inferiorly through the right atrium ultimately reach the AV node, which is normally the sole route of entry of the cardiac impulse to the ventricles. Atrioventricular conduction. As we know, the atrial excitation wave reaches the ventricles via the AV node. In adult humans, the node is approximately 22 mm long, 10 mm wide, and 3 mm thick. The node is situated posteriorly on the right side of the interatrial septum, near the ostium of the coronary sinus. The AV node contains the same two cell types as the SA node, but the round cells in the AV node are less abundant and the elongated cells prominent. The AV node is made up of three functional regions. First, the AN region, transitional zone between the atrium and the remainder of the node. Second, the N region, the mid portion of the AV node. And third, the NH region, the zone in which nodal fibers gradually merge with the bundle of His, which is the upper portion of the specialized conducting system for the ventricles. Normally, the AV node and bundle of His are the only pathways along which the cardiac impulse travel from atria to ventricles. Several features of AV conduction are of the physiological and clinical significance. The principal delay in the passage of the impulses from the atria to the ventricle occur in AN and N region of the AV node. The conduction velocity is actually less in the N region than in the AN region. However, the path length is substantially greater in the AN than in the N region. The conduction time through the AN and N zones account for the delay between the start of the P wave, that is the electrical manifestation of the spread of atrial excitation, and QRX complex, that is spread of ventricle excitation, on an ECG. Functionally, the delay between the atrial and ventricular excitation permits optimal ventricular filling during atrial contraction. Ventricular conduction. The bundle of His passes subendocardially down the right side of the interventricular septum for about 1 centimeter and then divide into the right and left bundle branches. The right bundle branch, which is direct continuation of the bundle of His, proceeds down the right side of the interventricular septum. The left bundle branch, which is considerably thicker than the right, arises almost perpendicularly from the bundle of His and perforates the interventricular septum. On the subendocardially surface of the left side of the interventricular septum, the left bundle branch splits into a thin anterior division and a thick posterior division. The right bundle branch and the two divisions of the left bundle branch ultimately subdivides into a complex network of conducting fibers called Purkinje fibers, which spread out over the subendocardial surfaces of both ventricles. Purkinje fibers have abundant, linearly arranged sarcomeres, as do myocytes. However, the T-tubule system is absent in the Purkinje fibers of many species, although it is well developed in the myocytes. Purkinje fibers are the broadest cell in the heart. They are 70 to 80 micron in diameter, compared with the diameter of 10 to 15 micrometer for ventricular myocytes. Particularly because their large diameter, conduction velocity is 1 to 4 meter per second in the Purkinje fibers exceeds that of any other fiber type within the heart. The increased conduction velocity permits a rapid activation of the entire endocardial surface of the ventricles. Impulse conduction in the right or left bundle branch or in either division of the left bundle branch may be impaired. Conduction block may develop in one or more of these conduction pathways as a consequence of coronary artery disease or degenerative processes associated with aging, and they give rise to characteristic ECG pattern. Block of either of the main bundle branches is known as a right or left bundle branch block. Block of either division of the left bundle branch is called left anterior or left posterior hemiblock.